Multiplex PCR. Multiplex PCR is a type of PCR which enables simultaneous amplification of many targets of interest in a single reaction. In a single PCR tube, we can amplify different regions of the genome. There are multiple primer pairs. Those primer pairs can be simultaneously used, and we can amplify different regions of the genome by using a single tube and by using a single PCR reaction. As a result of multiplex PCR, amplicons of different sizes are produced. Because we use multiple primer pairs, each primer pair produces a PCR product, which is also called as amplicon. So there can be different size of the amplicon, but there is also possibility that different amplicon can also be of same size. Here we can see that what is the concept of multiplex PCR. So this is the, let's say this is the DNA. In case of DNA or in case of the genome, we can see that this is the first pair of primer, this is the second pair of primer, third pair of primer, and fourth pair of primer. So each pair of primer will bind to specific locations on the genome and will amplify the region. So here we can see that as a result, this is amplicon 1, this is amplicon 2, amplicon 3, amplicon 4, and amplicon 5. So 5 amplicon can be produced, even more than 5. It can be 10 to 15, sometimes 10 to 15 amplicons are produced within a single PCR tube or within a single PCR reaction. So such a PCR in which we use multiple pair of primers and produce multiple amplicons, such a PCR is called as multiplex PCR. So in case of multiplex PCR, single reaction can produce many amplicons. Within a single reaction, there are multiple primer pairs, and as a result of the reaction, multiple amplicons can be produced. So while performing multiplex PCR, one has to optimize the annealing temperatures, because there are different set of primers and each set has different annealing temperature. Before performing the multiplex PCR, the condition of all the amplicons within a single PCR reaction have to be optimized. Amplicon size should be of different size or it can be labeled. While performing the multiplex PCR, if the primers they are not labeled, then the size of the amplicon should be different. While if the amplicons they are of overlapping size, then the primers they should be labeled. So as a result of labeling of the primers, we can identify a different amplicon of the which are of the same size by using the fluorescence. So labeled primer are used when the amplicons they are of overlapping size. Before performing this type of the PCR reaction, the conditions of the primers must be optimized before performing the reaction, because there are different primers and each pair of primer has different annealing temperature. So all the condition of all the primer pairs should be optimized. So we can say that multiplex PCR is such a PCR in which more than one pair of primers are used, multiple amplicons are produced by using multiple pair of primers. This type of the PCR is called as multiplex PCR.